name's Gregory McMurtrie. I'm a student at UBC, as kind of told you guys. I'm going to talk to you guys about the epitaxial growth of graphene by chemical vapor deposition. So, what's graphene? It's a two-dimensional, one-atom thick molecule comprised of a honeycomb lattice of carbon atoms, and it looks exactly like a honeycomb. That's why we call it that. It's very interesting. It's been theorized since the 60s. People have always known that it existed. It's graphite in your pencil. It's comprised of multiple layers of this, but it was only until 2004 that Andre Geim and Konstantin Nosilov were able to isolate graphene, and they called this technique mechanical exfoliation. And that's a pretty fancy word, but it really means getting a piece of Scotch tape and some graphite and sticking it to graphite over and over again, sticking the tape to itself, and putting it onto some silicon, and that works. But it's not a very consistent technique, and it doesn't work very good for large-scale production of graphene. So, one of the other disadvantages is that you get very small domain sizes. So that means each individual piece of graphene is only microns squared at most, and that's really not useful for any sort of scalable applications of graphene. And one of the other disadvantages is that it's very difficult to differentiate between the number of layers of graphene. Because being only one atom thick, it's very easy to see 2 atoms, 3 atoms, or few layer thick graphene and not be able to tell what you're getting. And different layer thickness seriously affects the electronic properties of your samples. So there's a technique called chemical vapor deposition, which is the epitaxial growth of graphene on a copper substrate, which allows for large domain sizes as large as the copper that you want to put inside your system. And I use methane as a carbon source, but many other sources have been used, like ethylene and polystyrene, actually, as well as some really odd ones like Girl Scout cookies and a cockroach leg, oddly enough. But yeah, you can pretty much make graphene out of anything with carbon. But what I did is CVD. So this is a SolidWorks model of the system that I built. On the right here, we have our gas sources. We've got argon for a purge line, methane for the carbon source, and hydrogen, which allows for the CVD reaction to be more of an equilibrium reaction, which I'll explain in just a second. So the carbon enters the furnace right there, as it's in the form of methane, and it forms CXHY species, which are what you need to form your graphene. So the hydrogen is removed from the carbon, and then it can grow on your copper surface, which is right about there. And so you're flowing your gas into your system, you heat the system up to 1,000 degrees Celsius, your carbon decomposes, and then you can grow graphene, which I think is pretty awesome. So on the right-hand side, next to the gases, you have mass flow controllers, which will regulate how much of the gases are going inside the furnace. And then downstream, we have an exhaust valve, which actually interfaces with a pressure gauge. And this allows you, with the combination of these 3 things, to finely tune the growth pressure and flow rates, which is critical when growing any sort of crystal. And this is all, there's a pump there you see, which keeps the whole system at a vacuum, so that you have precise control over the growth parameters. So in order to have, just go back here for a second, we've got the mass flow controllers over on the right, which interface with the control box. And then over on the left, we've got the pressure gauge and exhaust valve, which interface with a different control box. So in order to have these 2 things talk to each other and have a more precise control of the system, I wrote some LabVIEW code, which allows for any sort of graphene growth recipe to be introduced. And then you just press the grow button, and it is a completely automatic process. So you can do this during the nighttime. You can do this whenever. So it's convenient to have samples prepared in advance. And another cool thing about this program that I wrote is that it stores all of the growth parameters as well as a log of the system pressure and gas flow during growth, which allows you to specifically characterize your samples and then try and compare what different recipes will give you for different samples. And these recipes can actually be loaded back into the program as the log file, can be loaded back into the program as recipes, so that you can have a very repeatable system. So you can say, oh, look, there was a pressure spike at 3 minutes into growth, and then your samples have some abnormality. You can understand why that's happening, or at least have a better guess. And there's also a couple safety features that I implemented in the program, which, for example, monitor the pressure spikes if they go too quickly, indicating a leak in the system. It will completely shut down methane and hydrogen flow, which, when you're working at 1,000 degrees, is a good thing. Okay, what else have I done? Okay, so I transferred my samples from copper as they're grown onto silicon oxide. So to do this, we want to deposit polymer on top, and you can actually pattern this polymer using standard photolithography techniques. So you can get a silicon wafer and add like a photoresist, and you can pattern it just using standard, like well-developed techniques. And you can then pattern. This here, they've got a series of squares, but you can actually, you can do any sort of micron scale features, which isn't enough to make like a nanowire, for example, of graphene, but you can make any sort of array of CVD graphene that you want, which is convenient. And so after doing all this, to characterize my samples to see if they were actually any good, I did Raman spectroscopy, which relies on the inelastic scattering of monochromatic light, which is usually provided by a laser. And this gives you an individual fingerprint of a system based on its vibrational modes. So this essentially means that every single molecule has a unique fingerprint in Raman, which this is the first literature that was done on Raman on graphene. And you can see the difference between graphite, which is essentially many, many layers of graphene. And you see here there's two peaks named the G peak and the G prime peak. They were named because they were found in graphite. One is at 1,580 inverse centimeters, and the other one's at 2,700 100 centimeters, so it's just wave number, as people like to call it. So in graphene, you see the G peak is actually much shorter than the G prime peak, and that is because of the different vibrational modes introduced based on its lower dimensionality. Okay, so I did Raman spectroscopy, and the first one we found, I did it on copper before transferring it using the technique I described before. And you can see a very sharp G peak and a G prime peak. And then this small peak right here is actually called the D peak, which is a defect band, which tells you there's the defects inherent in the graphene, because it's not always a perfect honeycomb lattice. There's these defects that can be introduced, like heptagon, pentagon defects, which are somewhat resistive, as well as grain boundaries, which are highly resistive. And knowing this height of this defect band while doing transfer of your samples will allow you to know more about your system when you go to characterize it using other techniques. So after I had it on copper, I transferred it to silicon oxide. And you can see here the G peak was greatly reduced. And so when you're looking at this one, you would think, is this not graphite? Why is the G peak so tall? Because as you see here, the G peak is quite thin, graphite is very tall, but I actually found this, my hypothesis, that's due to coupling of the graphene with the copper substrate underneath, and introducing different vibrational modes into the system, which can inaccurately lead you to assume that you have multi-layer graphene instead of the samples that you want, which look like this. And this is actually the same sample after doing the previous Raman spectroscopy. And you can see that it's quite pristine graphene. And the defect peak for what we would call pristine graphene is when it is less than about a quarter of the height of the G prime band. And you can see here that the samples were awesome. So yeah, I think that's actually it. So
with your chemical vapor deposition technique, do you get higher or lower quality graphene than with the Scotch tape?
I'd say the exfoliated graphene is still probably the best samples that you can get as far as just mobilities, for example. And the only problem is that depending on your application, they're very, very limited. Like I'm working with a group that is making solar cells using Schottky junctions. And for that application, you need to have large sheets of graphene to use as an electrode. And as well as for like transparent touchscreens, for example, which is another application, you need to have much larger pieces than mechanical exfoliation can provide. But if you're just doing like transport measurements, mechanically exfoliated graphene is still your best choice.
So you mentioned that you have a small traditional method, and your method you roll the graphene through gauge flow. So do you have any idea about how large is your?
They're actually only limited by the size of the copper. So the only thing is they're not going to be a continuous domain. That's the main difference, because you're going to get multiple seed locations throughout the copper, and then they all grow to meet each other. The only problem is, depending on your application, these grain boundaries, you get a lot of heptagon-pentagon defects, so they're highly resistive, and you can't measure for like ballistic transport, for example, across a grain boundary. You need to, but yeah, you're really, it's unlimited size.
Have you looked into the ability to dope the graphene sheets?
I have not, but some people have been doing like interscalation of nitrogen, for example, and that can lead to a lot of interesting properties, but I personally have not. That's outside the scope of the research I've been doing.
Okay, do you know if that's inherent to CVD or just that you haven't heard anything about it?
Oh no, you can definitely dope during CVD. You can introduce more reagents during the growth process, and it's actually one of the big important things is to not do that when I'm growing my samples, because I'm trying to get pristine, just regular undoped graphene. So yeah.
This might be a silly question, but when you're transferring from copper to silicon, did you ever re-image the copper to see if all the graphene was transferred?
Well, the reason you can see graphene on silicon is actually due to an interference effect. You deposit a 300 nanometer silicon oxide layer on top of your silicon, and this causes an interference effect with your one atom thick graphene. You can't actually see graphene on copper. There's the only difference between a copper foil before you put it in and a copper foil after is that it won't oxidize for about 2 weeks after, so it's slightly shinier. But yeah, there is no actual way to tell if there's graphene on copper.
What ratio of gases did you use for this, and what different ratios of gases build different structures, or you have more defects depending on your ratios, or what?
I've actually done quite a bit of testing doing this, and there's quite a lot of literature about the ratio of hydrogen to methane, and what that really causes is the change between an equilibrium and a kinetic reaction. See, when you have when you have a lot more methane than your hydrogen, you're going to get these large dendritic non-hexagonal structures. They have six-fold symmetry, but they look like dendrites that are growing. And while the more hydrogen you have, you're going to etch off the less favorable growth directions, and you get more sharply edged hexagons. So so more hydrogen has been found to create better quality films, but the growth time has to be greatly extended in order for them to be completely continuous. So it's kind of a toss-up between continuity and quality, and that completely depends on your applications.
So, what are you going to do from now?
I want to do scanning electron microscopy, which that was the goal for the project. I kind of set a much loftier timeline than I was working in. I wanted to do deposition of potassium onto the graphene and do scanning electron microscopy, and then maybe even also doing like self-assembly on top of the graphene, because it works very well as like an easy planar surface that you can bias and do scanning electron microscopy, which is actually the main focus of one of the groups I was working with.
So my silly question is that where did you exactly do that? Because those three guys
Sarah does.
Sarah does?
Sarah
They are spread around campus.
Yeah, see, Josh Hope does a lot of the transport measurements. That's his thing. He does low temperature mobility measurements, while Sarah does the STM, and then Peyton Servati does the solar cells. So it's all over the place. I've worked with all three of them. This is actually the second CVD system I've built.
Yep. Good. Thank you, man.
Cool.
Good.
Just before everyone leaves, I have an announcement. Okay, since a lot of people are interested in going to the Atlas lecture, there are some room available for the tours. So if you want to go and you're interested, you can just show up at the lobby at the time that you're supposed to meet, and then see if there is a room for you. So for Large Bennett Telescope, it's at 1:15. For D-Wave, it will be at 1:45. And Boreal Genomics is at 2:15.